Okay, I'm just going to give this one to you all so we can go ahead and move on. But those checkpoints are there to prevent any mistakes from occurring in a new cell. So we need checkpoints to serve as protection from any mistakes taking place and from those mistakes being repeated and copied into a new cell. So if there are mistakes, then the cell decides, I'm not going to reproduce. I'm not going to copy that mistake, because then you end up with two cells with that same mistake. If those checkpoints are not there, then the cell can eventually become cancerous. And if it continues to reproduce with that same mistake, you can end up with a tumor. So we need the checkpoints to prevent mistakes from occurring. All right, but excellent. Good job there. So just as many notes as you all took yesterday, I want you to take just as many today. I want you to take good notes, effective and efficient notes. But I think today won't, I don't think today will take as long as yesterday did, but we're going to go ahead and jump into it. So go ahead and get your notebooks out if you don't have them out already, and be ready to write with me. As you all do that, I want to make you aware that starting today, September 15th, all the way until October 15th, we have National Hispanic Heritage Month. We're celebrating the history of all of the contributions made by people of Hispanic descent to our cultures, our country's culture, and our country's, well, all of the fields in our country. So it could be artistic, athletic, social, cultural, ethnic, culinary, academic. So in all the ways that Hispanic Americans have contributed to the enriched culture of the United States, we want to celebrate those contributions and the people who made them this month. And we start with September 15th because it is the anniversary. Today is the anniversary of independence for 5 Central American countries: Costa Rica, El Salvador, Guatemala, Honduras, and Nicaragua. And so these countries are celebrating their independence from Spain today, September 15th. And tomorrow, Mexico will celebrate its independence. However, Mexico did not win its independence on September 16th. They celebrate independence on the day that they began their fight for independence. So I've always thought that that was a fascinating fact, because Mexico chooses to celebrate the struggle for independence rather than the triumph of independence. And I think that that is very much so aligned with Mexican and Mexican American culture. And then Chile celebrates its independence this Friday on September 18th. So we've got a lot of important dates that take place between September 15th and October 15th. And so it made sense when this Heritage Month was signed into law by President Ronald Reagan in 1988. It made sense for those dates to be chosen. And just as another aside, and to provide you all a little bit more thoughts about National Hispanic Heritage Month, it's important to recognize that all of these different Hispanic and Latin cultures have slightly different cultural norms. They have different histories. They have different expectations. And even in the way that we refer to them, we have to be careful about the words that we use. So you've probably heard the word Latino, you've probably heard the word Hispanic, but what you should know is that those two words are not exactly the same. Latino refers specifically to people who are from Central and South America, whereas Hispanic refers specifically to people who are Spanish speaking or whose ancestors were Spanish speaking. And so Latino does not refer to Spain because Spain is a European country. Latino only refers to people who are located in Central and South America, or who are descended from Central and South Americans. So Latino can include Brazil and French Guiana and Guadeloupe and Haiti and Martinique and Saint Martin, because those countries are located in Central and South America, but they are not derived, or they are not descended from Spanish-speaking people. Brazilians speak Portuguese, which is not Spanish, so they are not Hispanic. French Guiana, French Guianese people speak French, so they are not descended from Spanish speakers, and so they are not considered Hispanic. And so what we have here is really a reminder of the many ways in which all of these South American and Central American and Spanish-speaking people are unique, and all of the different cultural influences that have made them who they are, from European influences, the history of colonization, indigenous histories, and as well as the history of African people who were brought over as a part of the slave trade. We see all of those cultures coming into play with many of these Latino and Hispanic cultures. And so it really creates a very unique and very blended society. And more recently, perhaps you've heard the term Latinx, which is used as a gender inclusive term, meaning that in Spanish and in many of the other languages that are spoken around the world, words are given genders. There is a masculine version of the word, or there is a feminine version of the word. For example, we can say Latino, which is more masculine, and then there's Latina, which is feminine. But Latinx is a new phrase that is used to describe people who don't want to be put into a box as being either masculine or feminine, or it's just being used to describe Latin people in general without giving it a gender. Some people do have a preference. Some people do prefer that you refer to them as a Latino or as Latino or Latina. And some people prefer that you stick with Latinx. So we have to be comfortable asking people, what is your preferred identity? And how would you prefer that I refer to you? There's nothing wrong with asking that question. And in fact, it can go a long way in making people feel welcomed and safe and seen in a space. So you all should be aware of these different terms. You should start thinking about them, especially as we continue our discussions of National Hispanic Heritage Month this month. And I'm going to be bringing in specific people and specific heritage and cultural facts into our classroom and related to biology. So be on the lookout for that. I'm excited about it, and I hope you are too. But we'll move on, and we'll start with our actual lesson of the day. We learned, and we reviewed it just now in the warm-up, that phase 1 is interphase. And this would be a great thing to write down in your notes to draw this general picture. It'll give you a good idea or a good starting point for the rest of our lesson and for the rest of the unit, really. So I would encourage you all to just draw a circle. It doesn't have to be a perfect circle. It could be an oval on your paper, and then divide it into these sections. But we know that the first phase of the cell cycle is interphase. But just like Jaden and Jessica and Nick told us during our review of the warm-up, interphase can be divided into 3 parts: gap 1, synthesis, and gap 2. Gap 1 is a period of growth. The cell is becoming a bit more sophisticated. It is producing proteins. It is making organelles. Synthesis is an extremely important step of the cell cycle because during synthesis, the DNA is replicated. If the DNA is not replicated properly during synthesis, then nothing that comes after is really going to work. Or if it does happen, then it's going to be damaging to the cell and to future cells. So we need synthesis to be carried out accurately. That's when the DNA is replicated. Then gap 2 is another period of growth. That's why I like to think of G1 and G2 as growth, the G standing for growth. Then we get to the second phase of the cell cycle, which is M phase. That's what we'll be talking about today. And our last phase of the cell cycle is cytokinesis. Cyto is a prefix, can also be a suffix, that means cell, and kinesis is a suffix meaning movement. So cytokinesis means cell movement, and it literally refers to how two cells will move apart from each other. They'll start off as one, and then they'll begin to move apart, create two identical cells. Okay. I'll leave this up for 10 more seconds. Again, I encourage you to consider drawing this or a similar version of it. Okay. So can anybody take themselves off of mute and answer this question? What do you think are some differences between sexual and asexual reproduction? What do you think are some differences between sexual and asexual reproduction? Anybody. What's the difference between sexual and asexual reproduction? I know you all have some thoughts. Just share with the class so we can all benefit from them.
So like ones that don't have have sex or something like that.
Okay. So yeah, essentially that's right. So sexual reproduction requires two organisms. You need one of the organisms needs to have a sperm and one of them needs to have an egg for sexual reproduction to take place, or the equivalent of those two things. With asexual reproduction, we only need one organism to do it. And essentially that one organism is just making a copy of itself. Okay, so there are different organisms like yeast and bacteria and some fungi that are able literally to just make copies of themselves. They don't require another organism to do that. But even most of the plants that we are aware of require two organisms. So in the early spring months, let's say in March and April, you might see a lot of pollen flying around, and it's kind of allergy season because there's a lot of pollen that floats around in the air. And that pollen is floating around because it's carrying seeds that are needed to germinate or fertilize other plants. So in some ways, that pollen is actually plant sperm. So even plants, and of course most animals, almost all animals and insects and birds and fish require two organisms to do sexual reproduction. So that's one of the differences between the two. And we're going to discuss more differences as we go throughout this unit. All right, so mitosis, M phase. This is where you all should definitely start writing. Remember, the things that are highlighted should be written down. Remember, before we get to M phase, we have to go through interphase. And during the synthesis portion of interphase, the DNA is replicated. So mitosis cannot happen until the DNA is replicated. That is very important to know. I'll say it one more time. Mitosis cannot happen until the DNA is replicated first. But once mitosis starts, it is the cell division process that makes somatic cells. So we talked yesterday about the difference between somatic cells and sex cells, or gametes. Somatic cells are your body cells, everything that make up your body, and then gametes are your sex cells, so either the sperm or the egg. Okay, and we can see a very basic animation of this concept in action. Concept of cell division. We can see that one cell is being split. Its nucleus is currently still connected. The cell membrane is currently still connected. But eventually it gets split so that we have two new daughter cells, each with their own cell membrane and each with their own nucleus. And here's another good example of that happening. So again, before mitosis can happen, we need to go through interphase so that we can replicate our DNA. If we don't replicate the DNA, we're in a bad position because, well, for reasons that we'll discuss soon. But we need to be able to replicate the DNA first. Now, we just talked about some of the differences between sexual and asexual reproduction. Mitosis is used for asexual reproduction. So cells are literally just making an identical copy of themselves. The two new daughter cells will have the same amount of DNA, the same amount of chromosomes. They will do the same things. All right. So during mitosis, cells undergo one round of cell division to make two identical diploid daughter cells. And I know that sounds like a lot, but we'll break it down. Just please make sure that you write down that sentence and write down the definition of a diploid. Okay. So mitosis undergoes one round of cell division to make two identical diploid daughter cells. So we're starting off with one parent cell, or one mother cell, as it says down here. And we're going to end up with two new cells. These two new cells are identical to one another. They are exactly the same. Same DNA. If everything goes right, we have the same DNA, we have the same function, we have the same structure. These cells are called diploid cells because they have one chromosome from each parent. They have paired chromosomes. So what we're going to end up seeing is that in the case of mitosis, the daughter cells end up with exactly the same amount of DNA as the mother cell. In order for that to happen, the DNA has to be replicated first. So interphase is still very, very important. Synthesis is still very important because we need to replicate the DNA before we can undergo cell division. And we can see cell division taking place here. The chromosomes are literally being pulled apart from the center, and they're forming two new cells. Okay. So mitosis, just like interphase, is made up of a couple of other phases, in this case, 4 phases. Mitosis is made up of prophase, which is first, then metaphase, then anaphase, and lastly, telophase. So please do write down the 4 phases of mitosis in whatever your preferred language is. And as you all think about practicing some good study skills and becoming strong students, one of the good tips that I used when I was a student in school was making acronyms for myself to try to remember as much as possible, especially if there was some sort of order to things like there is here. Prophase is first, metaphase is second, anaphase is third, telophase is fourth. That's a good habit to get into. So you might make an acronym for yourself. And my students in my first year teaching at West Meck, my biology students, they came up with the acronym to represent interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. So that's IPMATC. And their acronym was I play music at the club. IPMATC. I play music at the club. And you can come up with your own. There are plenty of other acronyms that you can use to remember these things and to remember their order, because that was still in order: interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. I play music at the club, same order. So it's just a good tip. It's a good strategy to help you remember things and the order in which they're written. Prophase, first step. And I know there's a lot here. Please, if you write nothing else down on this slide, please write down this first bullet point. The nuclear membrane dissolves during prophase. The nuclear membrane dissolves. And what that does is it allows access to the chromosomes that are inside of the nuclear membrane. Once that nuclear membrane dissolves, there's no longer any separation between the chromosomes and the rest of the cell. So now we have access to those chromosomes. Keep in mind, before this happens, we have to replicate the DNA. I know I keep saying it, but it's so important. Additionally, during prophase, the chromatin condenses into chromosomes, and the centrioles and the spindle fibers form. We'll talk more about that. The centrioles and the spindle fibers are essentially like a fishing rod. The spindle fibers are like the line on the fishing rod, and then the centrioles are like the axle on the fishing rod that allow you to reel it in. Okay. So prophase is the first step, and we can kind of remember that because pro is a prefix that means before. So it happens right before the cell is divided in two. Then metaphase. Meta is a prefix that means middle. And what happens is that the chromosomes literally line up in the middle of the cell. Chromosomes line up in the middle of the cell. They also attach to spindle fibers. Spindle fibers. Okay. Our third step is anaphase, and ana is a prefix that means apart. So the chromosomes literally get pulled apart. So I use my spindle fibers to cast out, and they get caught on to the center of the chromosomes. And then I reel that spindle fiber back in. My centriole reels the spindle fiber back in. And in doing so, I'm pulling apart the chromosomes. So if we imagine the chromosome as an X, once the centriole starts reeling the spindle fiber back in, it literally pulls the X apart, and both halves of the X move away from one another. So after the chromosomes have lined up in the middle during metaphase, they get pulled apart by the spindle fibers and the centrioles during anaphase. And then my last step is telophase. Telo is a prefix that means end. So telophase is the last step of mitosis. And during telophase, the cells begin to cleave, meaning they start to get cut off from one another. So what we're seeing is that new daughter cells are forming now. The chromosomes have been pulled apart. A new nucleus is going to form around those chromosomes, a new nuclear membrane. And then two cells are going to start to get pinched off from one another, and eventually we'll have cytokinesis as they move apart, as they move away from one another. Okay. And if you all need any more time on a slide, if I'm moving a little bit too quickly, please don't hesitate to, to tell me. Additionally, if you need to go back and see something that I went over, you can do two things. You could just ask, or you can make sure you check out the recorded version of the presentation after it's been uploaded. All right, so again, after telophase, we've got cytokinesis as the cells actually get separated from one another and they move apart. So this is another good image. If you have drawn the earlier image that I showed, then this probably is not going to be necessary for you. But if you didn't draw that earlier image, then this one might be a good one for you to have in your notes somewhere. It really shows each step. So we've got interphase, which is G1, S, and G2. Then we've got mitosis, which is prophase, metaphase, anaphase, and telophase. And then we've got cytokinesis, which is when the cell splits. So I'm leaving this up for the folks who might want to write it down. Okay. All right. So let's practice a little bit. What would be the correct order of cell division. If we had to reorder these pictures, which one should be first? Which picture represents, and I'll give you a hint, there are kind of two pictures of prophase here. So which one should be the first prophase picture? Remember,
2.
yes, good. Number 2 is the first image. So this is the first part of prophase. We can see that the centrioles are starting to form, the spindle fibers are starting to form, and the nuclear membrane is actually going to start to degrade. Oops. What would be the second picture?
Number 4.
Number 4, good. So the nuclear membrane has now degraded. If you can see here, it's a solid line. Here we have it represented as a dotted line, and we can see that the spindle fibers are starting to move into position. What comes after that? So we've got number 2, number 4, then what?
Number 3.
Number 3, excellent. So we can see that number 3 is metaphase. We can see that the chromosomes are lined up in the middle of the cell. So that's got to be metaphase. Meta means middle. Which one would be anaphase? We only have 2 left now.
Here's a bag.
1.
Turns around.
Excellent, thank you. Number 1 should be fourth because ana means apart. So we can literally see how these chromosomes that started off lined up in the middle during metaphase, they're getting pulled apart now during anaphase. So that leaves that number 5 is our last step. And you can see that now 2 cells starting to form. We've got another nuclear membrane that's starting to form. And there's another nuclear membrane in the other cell. And they're also starting to move away from one another. So good job. Really the last concept here, and the only thing that you should write down is this part. Chromosomes, the number of chromosomes stays the same from the parent to the daughter cell. And this is specifically from mitosis. The number of chromosomes remains the same from parent to daughter cell. So if my parent cell has 46 chromosomes, that means that my daughter cells should also have 46 chromosomes. And the reason for that is because of DNA replication. Between this step and this step, we can see we went from 46 chromosomes to 92 chromosomes. So it doubled, and that's because of DNA replication. We doubled, we copied our DNA. And that's because we want to be able to split it in half to end up with the original number. So first I need to double it, but then I split it in half to get back down to that original number. 46 gets doubled to 92. And then I cut it in half to go back down to 46. I start off with 46, I end up with 46. I start off with the diploid, I end up with a diploid. And this is specifically for mitosis. So if I have a banana which has 14 chromosomes, how many chromosomes should this step have? Remember, first I need to replicate my DNA. I need to double this number. I need to copy my DNA. So what number should be here? Hello? What number should go here, people? Oh, I see people are 28. Thank you, Jessica. Yeah, so I should have 28 right here. And then it gets cut in half again, so I should have 14. So I start with 14 and I end with 14. And in the middle, after my DNA has been replicated, I should have twice that number. So 14 to 28 and back down to 14. Just like over here, I had 46 to 92 and back down to 46. All right, so we've got 20 minutes. There are 2 assignments. There's an exit ticket and there's a mitosis practice assignment. The mitosis practice assignment is probably will take you about 12 minutes and the exit ticket is short, so it'll take you about 8 minutes. So you have enough time to do this before your 4th block class. So go ahead and knock it out. I will be here on mute if you need anything and just let me know. Okay, people, it is 1:20, so at this point, it's time for you to start getting ready for your 4th block class. I appreciate your engagement today, and I hope you can come back tomorrow with that same level of enthusiasm. If you need anything between now and then, please don't hesitate to reach out. I'll stop sharing my screen and stop recording, and I hope you have a good Tuesday.